Hey there, this is Dr. Allen. We're going to talk about protein synthesis in today's lesson. Here's the objectives. We did talk about transcription in a previous lesson, so we're going to focus more on translation and the roles especially of mRNA, tRNA, rRNA, as well as the triplets, codons, and anticodons. All right, here's a preview of what we're going to talk about today. All right, now previously we had looked at this portion right here, where we have the DNA that goes over to the mRNA. That's the transcription part. Remember, there is that genetic code. The DNA has G, C, T, and A, like so T, A, C, G, G, T, and it's this big long letter sequence in the DNA. This sequence is generally broken down into groups of three, and those are called triplets. The mRNA also have groups of three. Instead of triplets, they're called codons. Transfer RNA also has groups of three. Those are called anticodons. And then also on the transfer RNA is an amino acid. Okay, so by the end of this lesson, you should make good sense of this, what appears to be very complicated diagram. All right, let's get into this. So in DNA, we had mentioned that there are 4 nucleotide bases. Remember that? A, G, T, and C. They also have that deoxyribose and the phosphate attached to it, but all we really care about right now are the 4 bases, represented by these 4 letters, A, G, T, C. These could be considered the letters of the genetic alphabet. Each sequence of 3 bases is called a triplet, as we said on that previous slide. And so the triplets could be considered words, as an example. So if these are letters, A, G, T, and C, putting them together in various combinations will give you a word. Now, in a gene, you can have a long sequence of triplets, so a long sequence of words that eventually forms, in comparison, maybe a sentence. And that sentence specifies a sequence of amino acids in the protein. Okay, let's get into that a little bit more. So here is the image from that free textbook we talked about before, where you have DNA with a template strand. Through the course of transcription, it develops into RNA. The template strand T goes to A goes to U, C goes to G, G goes to C, right? And that's how I have it summarized over here, because remember, RNA does not have thymine. It does not have a letter T. It's been replaced by this letter U. Why is that? I can't tell you that. That's just the way that it is. Makes things a little more interesting, I guess, right? So T to A to U, C to G, and G to C. Those are the complementary pairs from RNA to that DNA template. Now we're going to talk about how we get from this RNA to the protein over these next few slides. This is a review from last week. We had the 3 types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. Before we get into all of that, I do want to take a little sidestep and talk a little bit more about exons and introns, because we're not going to get into too much detail in this course, but I think it's worthy for you to understand and see this, because you might hear this in future courses, and this will be maybe more of a memory jogger for you as well. But exons and introns, these are sequences on the DNA in the gene of the DNA, so sequences in the DNA gene that are either expressed or not expressed. Okay, exons are those portions of the gene that are expressed, and I underlined the EX because it's like exons. Exons are expressed. Introns, on the other hand, are intervening sequences between the exons, and these are not used. So within that particular gene, there's little gaps in there, these introns, that are not used at all for the purpose of protein synthesis. So the messenger RNA will go through here and it'll transcribe this template from the DNA, it'll end up being like this. They call it pre-mRNA, for example, right here. But because some sections are exons and some sections are introns, these little introns are spliced out. And so the actual final piece of mRNA looks like that. Okay, so the DNA is just longer than the actual final mRNA, which generates the protein, like so. So just an important concept for you to know, introns versus exons. All right, so let's go ahead and talk a little bit more about translation. This is from your text, Focus Figure 3.5. This will be taken into several steps. Now, in the nucleus, we talked about how this template strand of DNA has this pre-mRNA. The introns are trimmed out, and so then you have your final piece of mRNA just consisting of the exons of that DNA gene. Exits the nucleus through these nuclear pores, and then you have your messenger RNA. This messenger RNA will go to a ribosome. Now, a ribosome has two units. Remember, the ribosomes are made out of rRNA. Remember, they were made in the or at the nucleolus within the nucleus. These ribosomal RNA, small subunit and this large subunit, will then join together onto this piece of messenger RNA at the initiator site. Okay, this is the initiator site, and that is called a start sequence. This will be important. We'll talk about this here later on, but this will be the start sequence. This is the initiator site of this mRNA, and then what will happen is these little bits of transfer RNA brings along the amino acids that'll be used to create the protein. Right? So that's the initiation. Small subunit, large subunit joins together with messenger RNA at the start sequence, and then the transfer RNAs will start coming in and bringing in those amino acids. So after that happens, after that initial start sequence, this is the beginning of that amino acid right here, this methionine right here. This is the beginning here. As this ribosome moves along the messenger RNA, more transfer RNAs are brought in, and then during this process of elongation, a peptide bond will form as the amino acids are being built. So like here in this part here, this first one right here, this is proline here. You'll have this transfer RNA coming into the spot here. When it does that, it'll bond this amino acid to the chain. See how the chain now elongates? Because this transfer RNA brought in this one called leucine. It comes in here. It'll make that bond, and then now we have leucine attached to proline. Now, as that happens, the ribosome moves along the messenger RNA. That previous tRNA goes away. And then now we have this next site here where another transfer RNA is going to come in, bond the next amino acid, and then it's going to keep moving and moving and moving, right? And eventually, you see how this polypeptide chain keeps growing and growing and growing. These are all amino acids. This polypeptide chain keeps growing and growing because that ribosome keeps going along that messenger RNA until it gets to this thing called stop. Okay, once it reaches the stop, that tells the ribosome, okay, we're all done. And then the ribosome releases. See how it kind of goes away. And then the peptide chain, the polypeptide chain is released. So let's go a little bit more into the genetic code. I had mentioned before that on the messenger RNA, each of these little codons here will code for an amino acid. And there are 20 amino acids, but you see how many variations of letters there that can exist. 3-letter variations using C, G, A, and U. And so several of these combinations can code for one amino acid. So like this one right here, leucine, LEU. It could be UUA, UUG, CUU, CUC, CUA, and CUG. That's just one example. Here I have here for phenylalanine, it could be UUU or UUC. Isoleucine is that. This is that start sequence. Remember when I said at the very beginning there's a start codon? That's this one right here, AUG. And then there's 3 different ones that can code for a stop to terminate the sequence. Now a little bit about this table right here. This is taken out of your textbook as well. And there's a method for reading this table easily. So if, say, the mRNA codon is UUU, the first thing you do is you look at this left side right here. You see how there's a letter G, A, C, and U. So there's a U right here. Then you would go to the top one to see what the second one is, and this is a U. And then the third one we said was a U, and that kind of will bring us to this point right here, UUU, and that's phenylalanine. If the letters were, if we want to see AUC, see that? We go A, and then U is in the column, and then the C is over here. That just helps you to find the code for whichever amino acid it is. And so that's the way that you can read this table to identify which protein is associated with which mRNA codon. Confused about what all these little letters mean? Well, here's just kind of the abbreviation table here, just for your own reference. I'm not going to make you memorize this because that's what these reference charts are for. But like MET is methionine, PRO is proline, right? So ILE, isoleucine. Isoleucine right here. All right. Now I have been talking about this thing called an anticodon and tRNA. Here's a little bit closer look at a tRNA molecule. Okay, so it kind of has this thing right here. This is made out of RNA, this little strand here. There's a little binding site up here at this end for an amino acid. In this case, it's isoleucine, ILE. And then on the other hand, you have this. On the other end, you have this thing called an anticodon, and in this case, UAU. So if you have a tRNA molecule that has the anticodon of UAU, we know that that has the amino acid isoleucine. How does that apply to protein synthesis? Well, the anticodons are complementary to the messenger RNA codons. Okay, so AUA, if the messenger RNA says AUA, the transfer RNA molecule will have this little anticodon UAU because they match. Remember, A goes to U, A goes to U, A goes to U, just like that. Okay, that's that complementary pairing. So that's how messenger RNA will code for isoleucine as the amino acid. And this transfer RNA molecule is what brings it over to the messenger RNA at the ribosome. Let's take a look at another example. So glycine, the amino acid glycine. So the messenger RNA codes for glycine are here, GGU, GGC, GGA, GGG. And I kind of have this written right down here in this little bottom right corner. GGU, GGG, C, GGA, and GGG. Let's just take a look at this one, GGA. Now this is the messenger RNA codon, GGA. Okay. This is the setup right here. This strand up here is the DNA. That's the DNA sequence. CCT, as an example, on the DNA sequence, will code for GGA on the messenger RNA molecule. That's the big long strand. That's the transcription phase. CCT, because C is complementary to G, C goes to G, and then T goes to A. So the messenger RNA codon will read GGA. It's complementary to the DNA one. The anticodon for glycine we know is CCU. And I said that the anticodon and the transfer RNA, the anticodon is complementary to the messenger RNA codon. C goes to G, C goes to G. But remember, messenger or RNA does not have the letter T. It's been replaced by U, so A goes to U. This is the anticodon in the transfer RNA. That codes for the glycine. Remember, the glycine is, in this case, we were looking at was GGA. The GGA, this was the messenger RNA one right here. Now, big picture-wise, within the cell, you can have what's called a polyribosome. Okay, and this is an actual image of a polyribosome with a messenger RNA sequence creating some proteins. This is through an electron micrograph. Look at how powerful that is, 400,000 X. Now, let's kind of take a look at this right here. This is the start of that mRNA. Remember that start sequence. Ribosomal subunits will come into that little spot right there and then start making the polypeptide. But in this example, a polyribosome means that there's multiple ribosomes being used to generate this protein. So it's not always just one ribosome creating one protein. You can actually have multiple ribosomes coming along at various stages along the process. See, right, in this case right here, a total of 4 proteins are going to be made at this one time because the ribosomes will come on and then zip along, make this polypeptide, and then once it reaches that end spot, the ribosome will kind of break off. So that's a polyribosome and a polypeptide. And this is just an example here in this image, right? So each of these is a ribosome, and this is a strand of messenger RNA. And as those ribosomes are going along that strand, it's generating the protein. Okay, so remember when we talked about the rough endoplasmic reticulum? I said it was this membranous structure, part of that endomembrane system, and it's rough because there's ribosomes along the outside of it, and it's used for making proteins. So here's a diagrammatic depiction of that. Now this is not a polyribosome here. This is just one ribosome, but they're just kind of showing it in different steps in the sequence. Okay. So here's a ribosome on the messenger RNA. The messenger RNA will come through and start building this polypeptide chain. Okay, don't worry about the signal recognition particle. I'm not going to make you learn this processing part, but just know that the polypeptide is being developed and growing inside of this rough ER. Remember, this is the inside of the rough ER. This yellow stuff is the outside. As it goes along, it starts to get processed. Here's a little sugar group added onto it. That little SRP is kind of removed, and so now you have this polypeptide chain. And once that is made, once it goes through there and it finishes being made, it'll be sent off. It'll be pinched off in this thing called a transport vesicle. That transport vesicle will go where? It'll go to the Golgi apparatus, and then it'll undergo additional processing, and then the protein will be sent off to do whatever it is that protein is supposed to do. So that's what happens in the rough endoplasmic reticulum. So some of these ribosomes are on the outside of the cell doing this, but other times it's on the inside of the rough ER, and that protein is just sent off to the Golgi complex, Golgi apparatus. Okay, so here's a summary slide. So this is what we started with, and I said by the end of this lesson, you should be able to recognize all the steps in this process. So we'll go through it here. So remember, this is the DNA molecule here, okay? And there's a gene, one gene of that DNA molecule, and here's a little segment of that, and this is just the representation here. This here, so your DNA strand here. The triplets, TAC, GGT, AGC, GAT, and so on. The transcription phase, where the messenger RNA is made, has complementary base pair sequencing. And so T goes to A goes to U, because there's no T's in RNA. It's been replaced by a U. C goes to G goes to C, G to C, T to A, and so on and so forth. That's the messenger RNA. And instead of being called triplets, they're being called codons. Now, if you remember that one table I showed you earlier on, we said that AUG is the start sequence. So when it comes upon AUG, it will start the protein, and AUG also codes for the amino acid methionine, and so that'll be the start protein. I'm sorry, the start amino acid for the protein, AUG. The anticodon is complementary to the messenger RNA. So on the transfer RNA molecule, the anticodon is UAC, A to U, U to A, G to C. This is the transfer RNA molecule with methionine on it, right? So if you look at the whole sequence of DNA here, these letters, this is the messenger RNA sequence. The tRNAs with their anticodons are complementary to that. And so that's how we know that AUG codes for methionine, CCA codes for proline, so on and so on and so forth, until it gets to the stop codon. Now there are 3 different stop codons, but for simplicity's sake, I just copied over and showed you this one. UGA means stop. See this mRNA, UGA means stop. Once the ribosome hits the stop sequence, it stops the synthesis, and the protein is then, I'm sorry, the polypeptide is then sent off to do further processing to whatever it needs to be for the actual cell to use. All right, well, this is a complex topic. It might take you a few times to read through these sections in the book, watch these videos, explore other sources, and we'll see you at the next lesson.